Hello, and welcome back to part 2 of chapter 1, The Principles of Life. Last lecture we discussed what constitutes life and how living organisms require energy to maintain organization. In this lecture we're going to discuss genetics, evolution, and end with the scientific progress. So let's get started. First we're going to talk about the genome. The genome is the information encoded by an organism's genes. A gene is a specific sequence of DNA that encodes for making one or more specific proteins. We will learn more about DNA in future lectures. As you can see in this picture, a gene, which is a sequence of DNA, encodes information that produces proteins. Many of these proteins then go on to drive chemical reactions or produce structures within our bodies. All cells have the same genome, but specific cells have specific functions. Different types of cells, such as a nerve cell or an epithelial cell, express different parts of the genome. How cells control gene expression is a major focus of biological research. Mutation is a change in the nucleotide sequence within a gene. This often results in the protein being altered. Mutations may occur during DNA replication or be caused by radiation or a chemical. Most mutations are either harmful or have no effect. However, some may be beneficial and improve the function within the organism. Or even less likely, a mutation will turn your pet into a Ninja Turtle, as seen above. Complete genome sequencing have been completed for many organisms. These sequences can be used to study genetic basis of physical structure, inherited disease, or evolutionary relationships, as we saw last lecture with phylogenetic trees. Genomic sequencing has created the field of bioinformatics. Bioinformatics is the study to organize and process the immense amount of data resulting from genome sequencing. Here's our first progress check for this lecture. Take a minute or two to answer these questions and continue on through the lecture. Next topic we'll go over is evolution. Evolution is a cornerstone of biology that guides much research to this day. Evolution is a change in the genetic makeup of biological populations through time. A common set of evolutionary mechanisms apply to all organisms. For example, all organisms have DNA that can be changed. The picture on this slide illustrates the evolution of an early vertebrate, from a worm to a lancelet to fish-like organism at the top. This occurred due to changes in the DNA of the fish's ancestor. Constant change that occurs among populations give rise to all the diversity of life. Next, we will talk about how evolution occurs through natural selection and adaptations. Charles Darwin proposed that all living organisms are descended from a common ancestor by the mechanism of natural selection. Natural selection leads to adaptations. These adaptations can be structural, physiological, or behavioral traits that enhance an organism's chance of survival and reproduction. I'd like to pose a question here. What must an adaptation be in order to benefit an entire species? Anybody? Adaptations must be inheritable, meaning that they must be able to be passed on to offspring in order for evolution to occur. This is one of the key traits of evolution. We're going to look at a few different species of frogs and adaptations they have for their specific environment. By looking at these frogs, you can tell they are related. They are frogs after all. However, you look at their limbs, you can see that they are different. The ones on the left have no webbing between their toes, while the ones on the right do. This difference of limbs gives these species unique advantages within their environment. There are two types of explanations that researchers try to explain an adaptation with. They are called proximate and ultimate explanations. Proximate explanation explains how an adaptation works. Our frogs with no webbing in their limbs are able to stick to the surface of trees and have no need for webbing as their habitat is the tree. Webbed-limbed frogs help them swim in water. Again, approximate explanation explains how the adaptation works. In the ultimate explanation, look at what process led to the adaptations. This explanation looks at the frog's ancestors and what circumstances led to this adaptation. Was it a predator, food source, competition, or a little of all the above? An ultimate explanation would look into these factors. Next, we're going to talk about theories in science and how evolution can be measured. Theory in science is a body of scientific work in which rigorously tested, well-established facts and principles are used to make predictions about the natural world. In science, if something is a theory, it carries a lot of weight behind it. This brings us to the theory of evolution. This theory is a body of knowledge supported by facts. As a result of this evidence, we can understand the processes by which populations have changed and diversified over time and continue to evolve. Researchers have been able to measure and observe evolution in several ways. By observing the fossil record, they can observe changes in species over long periods of time. Historically, this is one of the main methods to measure evolution. With the advent of genetic testing, researchers can now look at the genetic composition of populations and measure changes over a shorter period of time with organisms such as bacteria and insects, especially as they become resistant to antibiotics and pesticides. Here's another progress check. Take a minute or two to answer these questions and continue on with the lecture. The rest of this chapter, we will talk about the scientific method and how scientists use it to guide inquiries. Scientific investigations are based on observations, experimentation, and reasoning. Biologists use these methods when studying the natural history of organisms. These observations and experiments often lead to more questions and avenues of research. You have probably heard about or have learned about the scientific method at one point in your life. This is one general way that biologists use to examine the natural world. There are some variations to the scientific method, but this slide shows the main parts of it. It usually starts with an observation, which can either be through the natural world or through a review of scientific literature. This observation can then lead to questions. These questions can lead to a hypothesis. Hypothesis is a statement that can be tested, that can answer your question. Your hypothesis leads to a prediction, which then can be verified using testing. We'll look at this a little more detail in the coming slides. This is a diagram of the scientific method. You start with an observation, ask a question, and make a hypothesis. From your hypothesis, you make a prediction and test your hypothesis. Your hypothesis is either supported or not supported. If it is not supported, maybe you need to change the way you're testing. Maybe you need to come up with a new prediction or a new hypothesis. Any result you get from an experiment, either in support of or refuting your hypothesis, is valuable. Many times, researchers will use results from a failed hypothesis to ask new questions, which can lead to a new hypothesis. Any significant results that support your hypothesis must be able to be repeated by other researchers, which leads to verification of your experiment. To answer a question, scientists use what is already known to form a possible reasonable answer, better known as a hypothesis. Hypotheses are not questions, but predictions. The hypothesis or prediction is made off of observations, and experiments are designed to test these predictions. The scientists in this cartoon are making a hypothesis, saying they shrunk or the spider grew. Now they need to form an experiment to see if their hypothesis is supported or refuted. Next, we are going to look at different kinds of scientific inquiry, what kind of experiments that they use. Controlled experiments are experiments where a variable is being investigated and manipulated. The variable that is being manipulated is one that causes the phenomenon being investigated. A method or experiment is conducted to manipulate only that variable in the experimental group. The results from the experimental group are compared to the control group. The control group does not have any variables manipulated. For example, in the picture above, you have two plots of dirt with a plant. You want to test fertilizer X. You give the experimental fertilizer to the experimental group on the left, not the group on the right. All other variables are treated the same, such as time, soil, and sunlight. Any different results between the two groups should be attributed to fertilizer X. Here's another example of a controlled experiment. Hypothesis is that exposure to atrazine, which is a herbicide, during larval development causes abnormalities in reproductive tissues of male frogs. 9 tanks were established, with 3 replicant tanks, each getting the same amount of atrazine. When the tadpoles transition into adults, the reproductive tissue examined for any abnormalities. At the conclusion of the experiment, data was analyzed. It was concluded that exposure to atrazine at concentration as low as 0.1 part per billionth induced abnormalities in the gonads of male frogs. You can see in the picture of the frog's testes on the right, there are eggs growing within them. Within experiments, there is usually an independent variable and dependent variable. The independent variable is what is being manipulated, and the dependent variable is a response that is being measured. Our previous example with the atrazine, the independent variable would be the amount of atrazine, and the dependent variable would be the amount of abnormalities in the male frog's gonads. On a graph, usually the independent variable is on the x-axis, the dependent variable is on the y-axis. Researchers are interested if there is a positive or negative response between the two. Such as you increase your independent variable, the dependent variable also increases or decreases. In Biolab, you'll have the chance to conduct an experiment and identify the dependent and independent variables. Next, we will talk about comparative experiments. These experiments, researchers look for differences between groups. With these, not all variables can be controlled. Perhaps none of them can. Data is usually gathered from different groups and compared using statistics. An example of this would be comparing atrazine between different lakes. You cannot control for temperature or size, or surrounding land use, but ideally you want your samples to be relatively similar. The hypothesis for this comparative experiment was the presence of atrazine in the environment, water, and how it correlated with gonad abnormalities in frog populations. 8 lakes were picked and sampled in the spring for frogs with their reproductive tissues harvested for analysis. Results from this comparative experiment indicated that atrazine levels above 0.2 parts per billionth affect male frogs' reproductive organs. This was not done in the controlled environment, but out in natural bodies of water. Note the difference between this comparative experiment with the controlled experiment from earlier. Next few slides are going to look at what science inquiry can and cannot do. Not all forms of inquiry into nature are scientific. In order to be a hypothesis, it must be testable and have the potential to be rejected. Science depends on evidence that can be quantifiable and reproducible. Religious or spiritual explanations of natural phenomena are not testable and therefore are not science. I want to stress this next point, because I don't want to hurt anyone's beliefs. Science does not say that untestable religious beliefs are wrong. It's just that they cannot be addressed using scientific methods. This is where philosophy and personal beliefs come in. They help to address issues of where we come from and is there a God? You just can't test it using the scientific method. Advances in science can raise ethical concerns, such as, should we clone? Should we genetically modify food? Just because we can do it, do we have the right to do it? Again, this is where ethics come in. Science does not tell us how to address these issues. I'd like to end the PowerPoint from this poorly drawn lines cartoon, where two scientists are debating about pushing the boundaries of science. Just because we can, should we? Who knows? This is what ethics is about. Why it's important for you to be an informed citizen and know what's going on with science, so you can make your own decisions. Last progress check. Please take time to answer these 3 questions. And with that, we conclude our first chapter. Thank you for listening, and have a good day.